These are the answers for the foundation exam questions for Purity and Chromatography. Question 1 asks us, some students tested a red cabbage leaf for starch. This is the method they used. Boil the leaf in ethanol, rinse the leaf in water and add the reagents to test the leaf for starch. Give one safety precaution the student should take in this test. The answer is wear safety goggles. Next question. Which reagent is used to test the boiled leaf for starch? It's iodine solution. Next question. What colour will be seen if the test for starch is positive? The answer is it turns blue-black. Next question. The students then used paper chromatography to investigate the coloured pigments in a red cabbage leaf. Complete the sentences. Choose answers from the box. So chromatography can be used to separate. It's a method of separation. Used to separate mixtures. In paper chromatography, the paper is part of the stationary phase, which means it doesn't move. The solvent is called the mobile phase. That means it does move. Next one. Table 1 shows the students' results. The distance each pigment moved was measured from the start line. So this is the distance moved in millimetres. The RF value is calculated using the equation. So the distance moved by the pigment divided by the distance moved by the solvent. The solvent moved 110 millimetres from the start line. Calculate RF value X, okay, in table 1, give your answer to two significant figures. So you'll do 17 divided by 110, and the answer is 0.15. Next question. The known ranges of RF values of some pigments are shown in table 2. So these are the known ranges. The RF value for the orange pigment in red cabbage leaves is 0.91. What is this orange pigment most likely to be? Well, the range only fits into carotene. Okay, so 0.91 only fits into this range, so it must be carotene. So you can see here the ranges, 0.89 to 0.98, 0.24 to 0.30, 0.20 to 0.26, 0.04 to 0.28, and you get 0.91 given to you, so therefore that fits into this range, so it must be carotene. Next one. The student used paper chromatography to identify the colours in a black ink. The diagram below shows the student's results. What colours are in the black ink? So this is the black ink. So what colours must be in the black ink? If we go across, we can see that red must be, green must not be there, and blue is. So the answer is red. The answers are red and blue. So we can see on the diagram, the match across, red is there, green isn't, and blue is, so blue and red must be present. There is one here though that's... Okay. There is an unknown. So that gets you 1 mark. But you need to say unknown for the other mark, because there is one that we don't know what colour it is. Next one, suggest which colour is the least soluble. So which is the least soluble? That means which one dissolves the least up the paper, and it's red. So the answer is red, the least soluble. The reason is because it travels the least far. Next one, use the diagram above to complete the table below. Distance moved by green, distance moved by solvent. Calculate the RF value for the green colour, use the equation. So if we go back, you can actually find out the answers using your ruler. So it travelled 12 millimetres and the actual solvent travelled 36 millimetres. So don't be afraid to get your ruler out and use the diagram. So that travelled 12 millimetres and this travelled 36. They do give you a range though, 10 to 14 and 35 to 36. So they do give you a bit of a range. So they give you a range here and they give you a range here for the distance that you've just measured using a ruler on your diagram. So the answer 12 divided by 36 equals 0.33. Again, they'll give you the answer if you've got between 10 and 14 and 35 and 36. So there is a bit of a range. Next one, the diagram shows a chromatogram for a food colouring. How does the chromatogram show that the food colouring is a mixture? The reason is that there is more than one dot in the vertical line. Obviously the horizontal line can have lots of dots. That don't mean it's a mixture, but this was all from one sample, so it's a mixture because it contains more than one dot. Again, a student makes the measurements for spot C. The table shows the results. Again, distance moved by spot, distance moved by solvent. So again, 7 divided by 39 gives us an answer of 0.1795, but it does ask you to give it to two significant figures, so read what it tells you. So obviously rounding up, the answer is 0.18. You'd be surprised how many people lose marks by not doing it to two significant figures. And don't forget that your significant figures is your 1 and your 8, not your 0. Plan a chromatography experiment to investigate the colours in an ink. So what you'd need to say to get the full 6 marks. So put dots of known colours and a dot of an unknown on a pencil line. Has to be pencil, so the line doesn't interfere with your results, doesn't travel up the paper. On the chromatography paper, then next place the bottom of the paper in water, which is your solvent, but making sure the start line is above the waterline. So again, the ink doesn't dissolve into the water. Leave the solvent to rise when the solvent is near top of the paper, remove and leave to dry, and then compare the positions of the dots for the known samples and those of the ink. This gets you 1 mark, this gets you 2 marks, 3 marks, 4 marks, 5 marks to say remove, and then 6 marks to actually say compare at the end. So you put the dots of the known colours and a dot of the ink on a pencil line on the bottom of the paper, roughly about 3 centimetres from the bottom. Then you place the bottom of the paper in the water, making sure that the start line is above the water. You leave the solvent, which is the water, to rise, and then when the solvent is near to the top of the paper, you remove the paper and leave the paper to dry. Then finally compare the positions of the dots for the known samples and those of the ink. So I hope you've done all right on that. Thanks for listening.